YouTube live class again for class 12 student. Today we will again discuss the same topic of translation while we haven't covered it up in the last class. So let me tell you what is translation, what we have discussed. Now translation is the process of synthesis of protein from an mRNA template. So this template helps in the synthesis of protein. How? It is being held by the genetic code. So myself Dr. S.S. SARDAR from RD Women's HSS Bhubaneswar. So today we are going to discuss the translation again, as we haven't covered it up. We only have covered it up to the step of initiation. So let's have a look. So before going to translation, we have discussed regarding genetic code. Now genetic code is a set of 3-letter word, or is a set of 3-letter combination of nucleotides which are called as codon, and each of which corresponds to a specific amino acid or to a stop codon. What we have discussed in the last class. So what we have discussed is, now because of the nature of genetic code and because of the genetic code only, the mRNA strand or the base sequences or the nucleotide sequences which are present on the mRNA strand, they are being coded into an amino acid as per the triplet codon. Okay. So this is the basis of genetic code, which is universal, which is non-ambiguous, which is commaless, non-overlapping, and it is degenerate, of course. So we have discussed this much in the last class. So the genetic code is a set of rules that defines how the 4-letter code of DNA is translated to the 20-letter code of amino acid. So we only have 4 nitrogenous bases or 4 nucleotides in DNA, which will be transcribed into mRNA, and we will have the same, not exactly the same, just the T will be replaced by U, but similarly we have 4-letter nitrogenous bases. So these are the 4-letter code. How this 4-letter code present in the mRNA will be transferred or will be translated into a 20-letter amino acid code. That is the magic of genetic code. So as all the base sequences or all the nucleotide sequences present in the mRNA strand, it will be read in the form of triplet codon, and each triplet codon, it codes for a single amino acid, and some of the triplet codon, they codes for stop code. So 3s are there. So what we have discussed, now the triplet codon has to be a 3-letter word, and this 3-letter word, the permutation combination gives rise to 64 numbers of codons which completely codes for 20 amino acids. So this out of this 64 codons, 61 codons it codes for amino acids, and 3 codons this acts as stop codon. So whatever sequence will be present in the mRNA transcript or in the mRNA template, now all will not be translated. We already have discussed, so all will not be translated, only a certain region of mRNA will be translated into a polypeptide sequence or into a polypeptide chain. So which part is that? That is the open reading frame, which starts from an initiation codon and it ends at a termination codon. So this is the boundary. It starts at initiation codon and stops at stop codon or termination codon, and within that region, that means from the initiation codon to the stop codon, whatever sequence will be there on the mRNA transcript, they will be translated into amino acid, or they will be coded into amino acid in the form of triplet codon. So this explains, or this is being explained by the genetic code. Now the historical background is, now it was first of all described by Francis Crick, later on by Nirenberg, Matthew, and which has performed many experiments which shows that the codons are triplet, and mostly they have taken a sequence like U, completely that U repeated many U's, and they they saw that triplet U it codes for phenylalanine. So, accordingly or after that, many of the experiments by Nirenberg, Leader and Hergovin Khorana, so all of them has worked a lot in completely establishing the genetic code and the codons that are present and which codes for the 20 amino acids. So, the features we already have discussed, that is it is universal, non-ambiguous, then it is non-overlapping, and most importantly these are degenerate. So, 64 codons are there, out of which 61 codons they code for 20 amino acids and 3 codons are there which codes for stop codon. So, this is all about the features. So, next, and this is the complete table of the genetic code, which tells you or which you can see it well that there are totally 64 codons like this, all these are 64, out of which you see the stop codons are like UAA. So, this is a stop codon UA, UAG and UGA. So, these 3 are stop codon, out of which, and the rest 61 they codes for certain amino acids. And here you can see, as I have already told you in the last class, that you see UUA, UUG, and this CUU, CUC, CUA, CUG, they all codes for leucine. So, you can see that many codons they code for the same amino acid. So, this explains the degenerate nature of genetic code. So, this is the basis. So, next we will go to our main topic, that is translation. Okay. Now, translation refers to the process of polymerization of amino acids to form a polypeptide. So, what is the process? This is a process of polymerization of amino acids into a polypeptide. So, all the processes like replication, transcription, translation, they are all polymerization reaction, but in replication there will be polymerization of polymerization of deoxyribonucleic acid sorry, whereas in case of transcription there will be polymerization of ribonucleic acid or ribonucleotides into a copy of ribonucleic acid, that is RNA. Similarly, translation is the polymerization of amino acids to form a polypeptide chain, and finally that polypeptide chain will undergo certain modification, certain post translational modifications to become a functional protein. And why protein is important, that all of us we know. A protein is required for all aspects of life, all metabolic activities of life. And for your kind information, all the enzymes are proteins. Very few like some RNAs and many ribonucleases and all. So, these are mostly some catalytic RNA, except that most of the enzymes they are proteins. Okay. And you know the enzymes they catalyzes all metabolic activities of our cell. So, the order and sequence of amino acids are defined by the sequence of bases in the mRNA, and the amino acids are joined by a bond which is known as peptide bond. We already have discussed this, that how a peptide bond is formed, that a peptide bond is formed between 2 consecutive amino acids. Now, the structure of amino acid we have discussed in the last class. Now, the structure is, there is a centrally placed, the structure is like there is a centrally placed carbon. Now, it is being what? This is being bonded to a carboxylic group, to an amino group, to an H atom and to an R1 group. If this is the amino acid 1, then we have another amino acid. Now, how the peptide bonds are formed? This is being formed between 2 consecutive amino acids. So, this is amino acid 2. So, with the removal of a water molecule, now there will be formation of a bond like this, and this bond is called as the peptide bond. So, this is our peptide bond that happens between 2 consecutive amino acids. In the upcoming slides, we will also visualize this. So, the formation of a peptide bond requires energy, and thus amino acids are activated in the presence of ATP and linked to their cognate tRNA by the process of charging of tRNA or amino acylation of tRNA. Now, what is how a peptide bond is formed? Obviously, any bond formation, it requires energy. Now, the energy requirement is fulfilled by the activation of amino acid over the charging of tRNAs. Now, I have already told you how an amino acid is activated. So, if charged tRNA are brought close enough to each other, a peptide bond is formed between them. So, a translation, okay. Now the mRNA transcript, it is formed from the 5 prime end to the 3 prime end. Now, it does not mean that all the mRNA will be transcribed, or all the mRNA will be translated into a polypeptide chain. Rather, there will be a certain portion which is the translated region, whereas the other regions are called as untranslated region. And the translated region has a boundary which starts with the initiation codon and ends at the stop codon or termination codon. And this boundary, within this boundary, whatever flanked regions are there, they will be translated into a polypeptide. So, what happens here? Now, here what happens? Now, this portion from initiation codon to stop codon or termination codon, this is called as, this region is called as open reading frame, or in other words, we also can call it as a sister. Okay. So, the open reading frame is nothing but this is a reading frame of what? This is a reading frame that is being recognized by the ribosomes as per the triplet codon, which starts with the initiation codon and stops at a termination codon. And as per the triplet codons, the ribosome goes on adding the amino acid by the help of the tRNA or the charged tRNA over the mRNA template. That means, what kind of amino acid or what types of amino acid will join one after another, that will be decided over the mRNA template. But the amino acid will be carried to that codon region by the help of the tRNA, and there will be a codon and anticodon interlocking. So, in this way, by the cumulative effect of mRNA, tRNA and rRNA present inside the ribosome, togetherly they help in the process of protein translation or simply protein synthesis. So, what are the reason? A translation unit, or this is also called as a translation unit. So, it is a unit which is present in mRNA in the sequence of RNA that is flanked by the start codon, as I already told you, and the stop codon, and codes for a polypeptide, and all rest of the region they are called as untranslated regions. So, mRNA has some additional sequences which are called as untranslated regions, and they are present before the initiation codon and after the stop codon. Okay. But they are also important in facilitating the process of translation. What does it mean? Now, certain portion, as I told you, the sign Dalgarno sequence which is present in this untranslated region before the initiation codon in case of prokaryotes helps in binding of the smaller ribosomal subunit to the mRNA strand. Okay. So, the first step, so the total process of translation can be divided into 3 steps. One is initiation, then elongation, then termination. So, initiation, elongation and termination are the 3 steps which is the exact process of translation. So, before that, there is an important step which is called as activation of amino acid. So, before the act of initiation, the amino acid has to be activated, and the tRNA has to be charged with the amino acids which has to come to the site of codons. Okay. And when the tRNAs are present on the different codons, now peptide bonds will be formed by the help of the larger ribosomal subunit, which has a catalytic enzymatic activity called as peptidyl transferase activity. So, let us have a look on the activation of amino acid. So, how the amino acids are activated? The activation of amino acid takes place in cytosol, obviously, because all the tRNAs which are required for carrying the respective amino acid to the site of codon, they are all present in the cytosol or cytoplasm. So, all the tRNAs, all the amino acids. So, now how many tRNAs are required? For 20 amino acids, so minimally 20 tRNAs should be there, but additionally there should be an initiator tRNA which should load the initiator your methionine, because initiation codon is always AUG and it codes for methionine. So, the initiator methionine or the methionine, in case of prokaryote it should be N formulated, or there would be another step known as formulation of methionine. Once the methionine is formulated, then only it will be loaded into the initiator tRNA. But in case of eukaryote, no such this formulation of methionine is required, but there is always a initiator tRNA which should be loaded or which should be come to the initiation codon and forms a codon anticodon interlocking, and it should carry the methionine at its respective 3' 8. Okay. So, the charging of amino acid, or sorry, the charging of tRNA is again otherwise is known as activation of amino acid. So, how it is being done? It is being done in 3 steps. Okay. So, first of all, ATP molecules binds to the amino acid. Then, so what is the enzyme which is being utilized here, or what is the enzyme that catalyzes this reaction? The total or the complete set of these 3 reactions are catalyzed by amino acyl tRNA synthetase. In the last class we already have discussed it. So, ATP plus amino acid, it forms a AMP amino acid complex with the removal of a pyrophosphate. Okay. Then the amino acid transferred to the enzyme. Then this AMP amino acid is transferred to this enzyme. Then it becomes, now this AMP complex, it is released and it becomes an enzyme amino acid complex, or the amino acid present within the enzymatic environment, and the AMP is being released. Now, here this is activation of amino acid. Amino acid is a neutral molecule. So, in order to form a peptide bond, it has to be activated. So, that activation is done by the help of ATP molecules. So, each amino acid is activated by a single molecule of ATP. So, next this enzyme again it forms another or it brings about the tRNA, and it joins the tRNA and amino acids together. So, a tRNA with the correct anticodon binds the enzyme, the amino acid is transferred to the tRNA and the enzyme is released. So, the enzyme holding the amino acid, now it tries to couple the tRNA with the amino acid. So, once the amino acyl tRNA is formed, the enzyme is released, and this is the process of activation of amino acid or charging of tRNA. Now, you see the tRNA is charged with the amino acyl portion. Okay. So, this is the diagrammatic picture. Now, you see this is the tRNA, and this always loads the amino acid towards the 3' end of the tRNA. Well, so the 3' end has an hydroxylic group obviously, and the amino acyl tRNA synthetase by the help of this enzyme. Now, this amino acid is loaded to this 3' end like this. Okay. So, now you see the tRNA without amino acids and the tRNA without amino acid, and this is the tRNA with amino acid. So, this is called as amino acyl tRNA, or what it is otherwise known as, this is otherwise known as charged tRNA. Okay. And this charged tRNA only will go to the site of codon, will form an codon anticodon interlocking, and the respective amino acid at this 3' end, they will be placed at the codon region. Okay. So, like this the translation happens. So, next come to the first step, that is initiation. So, initiation is the first step, but initiation in itself, now all the RNAs are required like an mRNA template, then your charged tRNAs, then your smaller and larger ribosomal subunit. So, all the RNAs are required, but apart from this, all the RNAs which affects the actual translation, but apart from this many translation initiation factors are required. So, in case of prokaryotes there are 3 initiation factors for translation, whereas in case of eukaryotes there are 9 initiation factors for the act of initiation. But more or less the mechanism of initiation is same both in case of prokaryotes and in case of eukaryotes. So, let us have a look at the prokaryotic initiation factors. Now, there are 3 initiation factors like IF1, IF2 and IF3. Okay. And the first step, the initiation factor IF3 binds to the 30S ribosomal subunit. Okay. The mRNA binds to the 30S ribosomal subunit in such a way that the first codon, that is AUG, that lie on the peptidyl site and the second codon lies on the amino acid site. Okay. So, the mRNA binds to the 30S ribosomal subunit in such a way, first of all the 30S ribosomal subunit, it binds to the initiation factor 3. Then the ribosomal subunit or the smaller ribosomal subunit, that is the 30S, it binds to the mRNA not at any position it likes, rather to a sequence which is present just before the initiation codon, which is called as the Sine-Dalgarno sequence. And this Sine-Dalgarno sequence has a complementary sequence which is or the Sine-Dalgarno sequence is complementary to the 30S ribosomal, to a smaller portion of the 30S ribosomal subunit, or the 16S rRNA present in the 30S ribosomal subunit, and with this interlocking, now the 30S ribosomal subunit will be placed in such a way that the first codon AUG, it occupies the P site of the ribosomal subunit, smaller ribosomal subunit, and the A site or the second codon, it occupies the A site of the smaller ribosomal subunit, that is the 30S or 40S in case of eukaryotes. So, the tRNA carrying, next what happens? So, we have now this is the picture. Now, you see this is the Sine-Dalgarno sequence, that is a purine rich sequence GGAGG like this. Now, the smaller ribosomal subunit, now this green portion, this is the smaller ribosomal subunit. Now, this has a sequence which is complementary to this sequence, Sine-Dalgarno sequence, and it is placed in such a way that the first codon, that is the start codon or AUG, this occupies the P site of the smaller ribosomal subunit. And if this is the second codon, it may be anything like NNN. So, we can call it as nucleotide, nucleotide, nucleotide. It may be any nitrogenous base as per the sequence the mRNA transcript has. So, this occupies this is the second codon and this occupies the A site. Now, as I already told you, now you see, now with the binding of the larger ribosomal subunit, now 3 sites will be created. One is E site, one is P site, another one is A site. E site is the exit site. Now, here always a pre-tRNA which is not having the amino acid being charged over it. Now, this will be left from this side. So, from the exit side that tRNA will be released. Now, the P site for the first time, or when the AUG codon is the first codon. So, for the first time the P site is occupied by the AUG codon, and it is otherwise known as peptidyl carrying site or peptidyl carrying site. So, that means it carries a long chain of polypeptide or peptidyl. While we will discuss elongation, I will tell you how this peptidyl group is being carried over the P site. Now, the A site, the amino acyl site, this is always a site which is called as amino acyl site this is always being opened for the binding or for attracting a fresh tRNA carrying an amino acid. Okay, so all the sites, so this is the incoming site for the tRNA carrying a single amino acid, it will bind to this site. Then the P site, it will always carry the peptidyl chain, but in the past instance, the P site is occupied by the AUG codon and it will only carry the formulated methionine or the methionine at the first place. Okay, and the exit site, it has the importance that once the peptidyl group is transferred to the A site, now this pre-tRNA will exit from the E site. Okay, so let us have a look at the initiation. Now how initiation begins. Now we are clear about the three sites E, P and A. Now the smaller ribosomal subunit has the P and A site which will be occupied by AUG codon for P and second codon for A. So here you see, now here this site is being blocked by the initiation factor 1. It is always blocked by the initiation factor 1. So you see, now the second part is that this IF2 or the tRNA carrying the formulated methionine is placed at the P site, no doubt. This specificity is induced by another factor called as IF2 with utilization of GTP molecule. The IF2 factor has a ZTPase activity. So a GTP molecule is carried over there, and whenever required, now because when there would be binding of the smaller ribosomal subunit to the larger ribosomal subunit, now this will be released, or with the hydrolysis of the GTP molecule, the GDP group is formed, GDP and PI, and this will release all the initiation factors, and only there will be an mRNA template, both the ribosomal subunit and the charged tRNA on the P site. So for that reason, IF1 blocks the amino acid site at the or just before the initiation. So if the IF1 prevents binding of FMET tRNA is in the A site. So why it is so? Because now you see, now IF3, now first of all the IF3 binds to this place, and it generally it is at the signed organosequence. It affects the binding of the signed organosequence into the 16 or the binding or just the interaction of this sequence with the 16S rRNA, 16S rRNA of the 30S ribosomal subunit. Then there will be placement of the smaller ribosomal subunit in such a way that the first codon is occupied by the AUG, the second codon is occupied by, the first codon is occupied by the P site and the second codon is occupied by A site. But there may be chances that this formulated methionine may block this place. So in order to prevent that, the IF1 initiation factor, it binds to the amino acid site. So that is why it blocks the incoming, the freshly coming tRNA or charged tRNA which may bind to the A site. So for the first instance, the amino acid tRNA carrying the formulated methionine will bind to the P site. So once everything is bind, next what happens? Now with the help of this IF2 factor which has a ZTPase activity and a GTP molecule is attached to the IF2 factor. Now this GTP molecule, it releases the energy for the dissociation of all the factors like IF1, IF2 and IF3. Okay. Now once every factor is being released, now the smaller ribosomal subunit carrying these sites will be joined by the larger ribosomal subunit, and this becomes the initiation complex, and this is the process of initiation. Now you see the A site is free. Now here you see the A site is now free. It is not blocked now. So now what happened? As per the second codon, now a freshly charged or freshly charged tRNA carrying the respective amino acid for the second codon. Let us suppose here we have U. So, what should be the amino acid? This should be the phenylalanine. So, the phenylalanine, now this will come. So, here the tRNA will carry a phenylalanine. Okay. So, there will be the formation of a peptide bond in the process of elongation. So, this is the concept that after the formation of initiation complex only, now it will proceed to the step of elongation. Okay. So, and the initiation complex is being held by, or the step of initiation is being held by 3 factors like IF1, IF2 and IF3 in case of prokaryotes, and 9 factors in case of eukaryotes, but the mechanism is almost similar. So, this is the formation of initiation complex, and you see there is a free amino acid site which is ready, and here the IF1 does not bind here, all is being released. So, now it is ready to bind a tRNA, freshly incoming tRNA molecule being loaded with the respective amino acid as per the second codon. Okay. So, these are the sequence how this factors bind. So, first of all the IF3 factor binds to this smaller ribosomal subunit, then the initiator tRNA comes and bind with this IF2 factor. Now, with the release of the GTP molecule, and here the GTP is hydrolyzed with this release of all the factors. Now, you see the IF1, it has blocked the second codon, and with the release of everything, now the larger ribosomal subunit comes and bind to this place. So, here it is the complete initiation complex, which is the 7Ts initiation complex in case of prokaryotes, 8Ts in case of eukaryotes. So, next, now this is the prominent diagram of the initiation complex in case of prokaryote. Now, you see how they bind. Okay. So, here it is IF1, IF3 and IF2. Now, they bind togetherly, and with the release of all these factors, now this binds to this and it forms the 7Ts initiation complex. So, next we will go to elongation. Now, elongation marks again 3 sub steps. Okay. First one is the binding of amino acid tRNA at the A site, second one is the peptide bond formation, and third one is the ribosome translocation. Okay. So, we have 3 sub steps in elongation. First one is the binding of amino acyl tRNA complex at the A site. So, how the amino acyl tRNA binds to the A site? Now, the second tRNA carrying the next amino acid comes to the A site, as I already told you. Now, this is the case. Now, here the second amino acid will come with the tRNA and will bind to this place. So, and recognizes the codon on the mRNA. Okay. This binding is facilitated by a elongation factor called as EFTU. Now, the elongation is affected by 2 elongation factors, one is EFTU and another one is EFGS. Okay. And there is another EFTS which helps in the cycling of this one, and it becomes EFTU. Well, so the EFTU, and it utilizes a GTP molecule. Now, after binding, the GTP is hydrolyzed and EFTU GTP is released, and this then enters into a EFTS cycle, as I told you, and it again becomes the EFTU. Okay. So, it becomes the EFTU. So, the binding of amino acid tRNA is like such. So, this is the first complex, or the it may be 7Ts initiation complex or 8Ts initiation complex. So, once this is being formed, now you see the second codon is free now, or here the amino acyl end or the A site, this is free now. So, tRNA being loaded with an amino acid will come and will bind to this place. Now you see, so when it will bind to this place, let us suppose at the second place, now it will bind. So this is something like this. So this is the tRNA carrying, let us suppose it is the tryptophan. Okay. So what will happen, there will be the formation of a peptide bond next. So once a peptide bond is formed, so it forms a dipeptide. So here what will happen, now this methionine will come to this site. Okay. So here the P site will be converted to an E site and the A site will be converted to a P site with the process of translocation. So what will happen, so all the time the P site will carry the peptidyl chain. Now here this is tryptophan methionine, next let us suppose the phenylalanine will join, next suppose aspartic acid will join. So likewise the chain extends and always the A site is free to what, to attract an incoming charged tRNA being forming an codon anticodon interlocking. Okay. So the peptide bond is, so next once they are being positioned, so the first P site we have methionine, at the second we may have any amino acid. So there will be formation of a peptide bond between these two and it is catalyzed by the peptidyl transferase enzymatic activity which is present in the ribosomal subunit. And which ribosomal subunit? This is the larger ribosomal subunit. So this catalyzes the peptidyl transferase activity. Now the tRNA at P sites becomes uncharged. Okay. So here you see now two amino acids are there. Now when they will, this is AUG, first amino acid or methionine, this is UCC and this codes for serine. So what will happen, this methionine will come and will bind to this place and this will form a dipeptide. So this is the first step of elongation and elongation takes place in cycles. That means a single peptide or first of all there will be joining of an amino acyl tRNA to the A site, then there will be formation of a peptide bond, then there will be translocation of ribosome and it takes place from one codon to another codon and it repeats in cycles. Okay. So each time there will be binding of an amino acyl tRNA to the A site, next there will be peptide bond formation between the consecutive peptidyl chain and the amino acyl or amino acyl group, then there will be translocation of the what, ribosomes just by a single codon. Okay. So after this dipeptide formation, now you see next this third codon is ready to catch a freshly incoming charged tRNA being loaded with its respective amino acid. Okay. So this is the peptide bond formation and you see this is a dipeptide bond formation. That means if it is methionine, if it is serine, now you see now this dipeptide is carried over the A site now. Then what happens? Translocation of this ribosome happens by a single codon. Now next is this one, this may be N, N and N. So by a single codon it translocates and what happens? Now this is the P site, this is the A site. So what happens? The P site is converted to the E site and the A site is converted to a P site and always a freshly A site is created. Okay. So this will happen because of ribosomal translocation. So what are the steps of ribosomal translocation? Let us see. After peptide bond formation, now the ribosome moves one codon ahead. It doesn't that it will jump over codon and codon. So it will just slowly slide one codon ahead. Okay. So that the codon which codes for a specific amino acid, it will bring the respective or the necessary tRNA and there will be a peptide bond formation between this dipeptide and the amino acid and a tripeptide will be formed. Next, this tripeptidyl carrying tRNA will be on the P site, and again freshly A site will be created. It will bring off another amino acid and there will be formation of a tetrapeptide or a tetrapeptidyl group will be carried over the amino acid. Then there will be translocation by another codon. Then this P site is converted to E, A site is converted to P, and a freshly A site is created. Okay. So what happens here after peptide bond formation? Ribosome moves one codon ahead along the 5' to 3' direction on the mRNA, so that dipeptide tRNA appears on the P site. Now actually the dipeptide tRNA was present on the A site, but because of the translocation, now the P site comes to the E site and the A site comes to the P site. So it appears on the P site and next codon appears on the A site, as I told you. The uncharged tRNA exit from the ribosome and enter into the cytosol through the E site. Okay. Now the ribosomal translocation requires a factor called as EFG-GTP translocase enzyme, which changes the 3D structure of ribosome and catalyzes the 5' to 3' movement. The codon and A site is now recognized by other amino acid tRNA as in previous. So when there is a fresh A site, it will be recognized by another amino acid tRNA molecule, okay, or complex. So the dipeptide on P site is transferred to the A site as we just now discussed, forming a tripeptide, and the process continues giving a long polypeptide chain of amino acids. So here you see, now for the very beginning there is E, P and A site. Now when the dipeptide is formed and it is being carried over the amino acid side, now with the help of this ZTP or this group, that is EFG-GTP transferase molecule or this complex, now you see this site, it is being translocated in this way, in an inclined way, and the 3D structure is changed. Now what happens? Now the P site is converted to the E site, A site is translocated to the P site, and always now you see a freshly A site is created which is free, and it is going to carry or it is going to attract another amino acid tRNA as per the respective codon. So in this way your translocation happens, and it always what? It always convert a P site to a E site and a A site to P site, and always a fresh A site is created. Fresh A site is created. How? By the ribosomal translocation. Okay. So the E site, from when, what happened to this E site? Now in the E site the free tRNA will come out to the cytosol. Okay. So this is about the elongation process. So what is there? What are the 3 sub-steps of elongation which happens in cycles? First one is the binding of the amino acid tRNA to the fresh A site. Next is the peptide bond formation. Next is the translocation of ribosome by a single codon. So how long does it take? It takes when or until and unless it reaches to the stop codon. So within the initiation codon to stop codon, if there would be 10 amino acid or 20 amino acid, now after there are 20 elongation steps or 19 elongation steps, now after that only it will come and reach the what? Stop codon, and it proceeds to the step of termination. So termination marks when the ribosome translocates and it meets the stop codon like UAA, UAG or UGA. So the peptide bond formation and elongation of polypeptide continues until the stop codon appears on the A site. So obviously the stop codon, again it has to appear on the fresh A site, but the stop codon is not recognized by any charged tRNA. So what happens? So as it is not recognized by any Cas tRNA, rather it is recognized by some releasing factor or by some termination factor, we may call it as. Now in case of prokaryotes, there are 3 releasing factors. One is RF1, one is RF2 and another is RF3, but in case of eukaryote there is a single releasing factor which is performing all the function which is being performed by RF1, RF2 and RF3 in case of prokaryotes. So if stop codon appears on the A side, it is not recognized by the tRNA carrying amino acid because stop codon do not have anticodon on mRNA. So the stop codon are recognized by the next protein called the releasing factor, we just discussed now. Now this is RF1, RF2 and RF3 in case of prokaryote, and the RF1, RF2 they recognizes the stop codon like UAA, UAG and UGA, whereas the RF3 releasing factor, it actually releases the peptidyl chain, and it releases the peptidyl chain into a polypeptide which will come out of the ribosomal site or from the ribosomal environment, and everything is get dissociated. So this all the 3 togetherly hydrolyzes and cause the release of all components like this smaller ribosomal subunit, larger ribosomal subunit, mRNA and polypeptide, all they separates from each other and the process of translation stops there. So what is the role of RF1? The RF1 recognizes the UAA and UAG, while the RF2 recognizes UAA and UGA stop codon. So the recognition part is being done by RF1 and RF2, and the releasing part is done by RF3 releasing factor, and everything is dissociated like mRNA strand, smaller ribosomal subunit, larger ribosomal subunit and the total peptide chain or polypeptide chain. Okay. So how it is done, let us have a look. So as I told you, in case of eukaryotes there is only a single releasing factor called as ERF, and this causes the dissociation and all the function being performed by RF1, RF2 and RF3. In case of prokaryotes, it is singly being done by the eukaryotic releasing factor, that is ERF. Okay. So let us have a look on the diagram. Now this is the stop codon. So the releasing factor, so here this is the peptidyl chain and a freshly A site is created where either UAG, UA or UGA is being now present. So when this is being present, it is not being recognized by any such tRNA. So here the peptidyl chain is being carried over the P site. Now the A site, as it does not have any tRNA, it is being recognized and being occupied by the releasing factor. So the release factor, it occupies the stop codon site, and the RF3 or the hydrolysis of the polypeptide, this peptidyl group is done by the releasing factor, and you see everything is dissociated. Now the mRNA, the larger ribosomal subunit, the smaller ribosomal subunit and the releasing factor, and it releases a free polypeptide chain, and this marks the process or the step of termination. So this is all about the different steps of translation like initiation, elongation and termination. So this is all about translation. Now here you may see that the RF1, RF2 they bind to this A site. This is a better clear picture. Now the RF3 bind and it releases with the help of ZTP hydrolysis, it releases all other factors. And this is a combining picture of initiation, elongation and termination. Now you see the initiation is being done with the help of this tRNA with first amino acid, and the anticodons should be UAC, obviously it will come and bind to this place, and here first of all and initiation complex is formed. This marks the initiation. Then elongation marks the first peptide or the first placement of the second amino acid or second charged tRNA carrying the second amino acid as per the second codon of the mRNA. So there will be formation of a dipeptide, After that the ribosome will translocate a single codon, converting the P site to E site, the S site to P site, and a fresh S site is created. So, in this way a long polypeptide chain is created in each cycle of elongation, and a single step of elongation marks the addition of a single amino acid, and it elongates the chain one after another by a single amino acid till it reaches the stop codon. So, the stop codon here, when the A site is being occupied by a stop codon, now here instead of the incoming or freshly incoming tRNA charged molecule or tRNA loading the amino acid, instead of that now the release factor will come and bind to the A site. So, as there is no such amino acid that has to bind to the peptidyl chain, or the peptidyl chain cannot elongate further. So, this release factor, it hydrolyzes the what this or it cuts this polypeptide chain and everything is being released. So, this is all about initiation, elongation and termination of translation, and after translation there is certain modification of this polypeptide chain occurs. So, this is called as post translational modification. It does not mean that a polypeptide is formed means that it will be functional. So, from a polypeptide to a functional protein, now it must undergo certain kind of modification, and this is called as post translational modification. So, many proteins undergo either a chemical modification at certain amino acid residues following translation. So, that is why this is called as post translational modification, and this modification are essential for normal functioning of the protein and carried out by one or more enzymes catalyzed reaction. So, that means let us suppose this is a polypeptide chain carrying many amino acids. Now, this polypeptide chain from amino group N to carboxylic group N, now this will undergo certain modification and it will become a functional polypeptide. So, a functional protein. So, how a polypeptide is converted to a functional protein, that is being explained by the post translational modification. So, this is all about translation and post translational modification. Thank you, and in the next class we will cover the last portion, that is gene expression.
No.